Hello everyone! This section is for bacterial cell wall. After learning this section, you should be able to describe peptidoglycan structure and compare and contrast the cell walls of typical gram-positive and gram-negative bacteria. Firstly, let's see the function of cell wall. The cell wall is a layer that lies just outside plasma membrane. It is one of the most important structures for several reasons. First, it helps maintain cell shape and protect the cell from osmotic lysis. Second, it can protect the cell from toxic substances. Third, in pathogens, it can contribute to pathogenicity. Cell walls are so important that most bacteria have them. Therefore, it is important to understand cell wall structure. After Christian Gram developed the Gram stain in 1884, it soon became evident that most bacteria could be divided into two major groups based on their response to the Gram staining. Gram-positive bacteria stained purple, whereas negative bacteria were pink or red. The true structural differences between these two groups did not become clear until the advent of the transmission electron microscope. More recent studies of diverse groups of bacteria have shown that these models do not hold true for all bacteria. Because of the ongoing discussions related to these new studies, we will refer to bacteria that fit the models as being typical gram-positive or typical gram-negative bacteria. Firstly, let's see the cell envelopes of gram-positive bacteria. The cell walls of Bacillus subtilis and many other typical gram-positive bacteria consist of a single 20 to 80 nanometer thick homogeneous layer of peptidoglycan lying outside plasma membrane. Peptidoglycan, also called murin. See the transmission electron microscope picture on the left. You can see the thin brown layer is peptidoglycan. Also, murin is M layer. The red line is plasma membrane, the PM layer. The space between M layer and PM layer is a narrow space. We call it periplasmic space, marked P. In contrast, the cell walls of E. coli and many other typical gram-negative bacteria have two distinct layers. See the picture. There is a 2 to 7 nanometer thick peptidoglycan layer, covered by a 7 to 8 nanometer thick outer membrane. One important feature seen in typical gram-negative bacteria is the periplasmic space. It is much wider than that in gram-positive strain. The substance that occupies the periplasmic space is the periplasma. See the transmission electron microscope picture on the left. You can see the periplasmic space is wider than that in typical gram-positive bacteria. Then let's see the peptidoglycan structure. Peptidoglycan is composed of many identical subunits. Each subunit contains two sugar derivatives. The first is an acetylglucosamine and an acetamuramic acid. We also call them NAG and NAM. There are also several different amino acids. Many bacteria cross-link the strands by connecting the carboxyl group of the D-alanine at position 4 directly to the amino group of diaminopalmolic acid, position 3 of the other strand. The position 5 D-alanine is removed as a cross-link is formed. Other bacteria use a peptide interbridge instead. You can see that on the picture on your right. So let's see the characteristics of peptidoglycan layer. The peptidoglycan layer is strong but elastic. It is able to stretch and contract in response to osmotic pressure. And this layer is also rather porous, allowing globular protein having a molecular weight as large as 50,000 to pass through. Variants of peptidoglycan are often characteristic of particular groups. After knowing the structure of peptidoglycan, let's see the typical gram-positive cell walls. Gram-positive bacteria have zinc cell walls, composed of peptidoglycan and large amounts of other polymers, such as teichoic acids. Teichoic acids are polymers of glycerol or ribitol, joined by phosphate groups. The teichoic acids are covalently connected to peptidoglycan or to plasma membrane. In the latter case, they are called lipoteichoic acids because they connect to the plasma membrane lipids. You can see the teichoic acid on the picture. The function of teichoic acids is unclear, despite the fact that they may constitute up to 50% of the mass of the cell wall. The periplasmic space lies between the plasma membrane and the cell wall. However, it is so narrow that it is often not visible. The periplasma has relatively few proteins. Then let's see the typical gram-negative cell walls. The peptidoglycan layer is very thin, about 2 to 7 nanometers depending on the bacteria, and sits within the periplasmic space. The periplasmic space is usually 30 to 70 nanometers wide. There are many enzymes and proteins in the periplasmic space of the gram-negative cell wall. The outer membrane lies outside the thin peptidoglycan layer. It is linked to the cell by bronze lipoprotein. You can see the bronze lipoprotein on the picture. It is the most abundant protein in the outer membrane. This small lipoprotein is covalently joined to the underlying peptidoglycan and is embedded in the outer membrane by its hydrophobic end. Possibly, the most unusual constituents of the outer membrane are its lipopolysaccharides, LPS for short. See the red layer. The lipopolysaccharides layer consists of 3 parts: lipid A, the copolysaccharide, and the oxygen side chain. The LPS from Salmonella has been studied most. The lipid A region contains two glucosamine sugar derivatives, each with 3 fatty acids and phosphate or pyrophosphate attached. The fatty acids of lipid A are embedded in the outer membrane, while the remainder of the LPS molecule projects from the surface. The copolysaccharide is separated into outer core and inner core, and joined to lipid A, and is structured of 10 sugars, many of them unusual in structure. The oxygen side chain or oxygen antigen is a polysaccharide chain extending outward from the core. It has several peculiar sugars and varies in composition between bacterial strains. So that's the detail of LPS. LPS has many important functions. First, it contributes to the negative charge on the bacterial surface. Second, it helps stabilize outer membrane structure. Third, it helps create a permeability barrier. Fourth, it plays a role in protecting pathogenic bacteria from host defenses. The last, the lipid A portion of LPS can act as a toxin and is called endotoxin. At last, let's talk about pore importance. You can see the pore importance on the picture. The presence of pore importance permits the passage of small molecules such as glucose and other monosaccharides, and this makes the outer membrane is more permeable than plasma membrane. So they are very important. Okay, that's all for this section. Here is what is for you.